On day 22, development of eye begins. From the lateral aspect of the forebrain, an ectodermal diverticulum grows out laterally towards the side of the head. Its distal portion becomes dilated to form an optic vesicle, and its proximal portion becomes constricted to form optic stalk. At the same time, from the surface ectoderm, a small area overlying the optic vesicle thickens and forms lens placode, which invaginates and sinks below the surface ectoderm to form the lens vesicle. The optic vesicle invaginates to form a double-layered optic cup. The inferior edge of the optic cup is deficient, and this notch is continuous with a groove on the inferior aspect of the optic stalk called optic fissure or the choroidal fissure. Vascular mesenchyme now grows into the optic fissure and takes with it the hyaloid artery. Growth of the margins of the fissure around the artery occurs and the fissure closes by 7th week. Failure of the fissure to close completely leads to coloboma formation, which may include the eyelid, pupil, ciliary body, choroid or optic nerve. By the 5th week, the lens vesicle loses contact with the surface ectoderm and lies within the mouth of the optic cup, the edges of which form the future pupil. Hyaloid artery is a branch of ophthalmic artery. It supplies the inner layer of optic cup, mesenchyme of optic cup and lens vesicle. The hyaloid vessels are enclosed in the closing optic fissure. Its distal portion will degenerate and proximal portion persists as central retinal artery and central retinal vein. Persistent hyaloid artery is when there is a persistent small part of the distal portion that is present as a freely mobile, non-functioning cord projecting from the optic disc into the vitreous. Hyaloid cyst may be formed. The entire distal portion may be persisting, extending from optic disc to the lens where the eye is usually microphthalmic. Next we'll discuss development of lens. It is formed from surface ectoderm. Steps of development are induced by signals produced by the optic vesicle. The thickening of surface ectoderm overlying the optic vesicle, known as lens placode, invaginates and sinks below the surface ectoderm to form the lens vesicles, which consists of a central cavity surrounded by a single layer of cells. It is covered by a basal lamina, surrounded by vascular membrane. Cells in this lens vesicle differentiation depending on their place. At the anterior wall of the vesicle is formed of cuboidal epithelium, which forms the anterior lens epithelium. At the posterior wall of the vesicle is formed of tall columnar cells, which rapidly elongate and become filled with proteins called crystallins. These crystallin make them transparent. The base of each primary lens fiber remains attached to the basal lamina posteriorly, while the apices grow forward towards the anterior lens epithelium, and the cavity of the lens vesicle gradually becomes obliterated. The nuclei of the lens fibers move anteriorly within the cells to form a line convex forward called the nuclear bow. Later the nuclei disappear. At the equatorial zone, anterior epithelial cells, the cuboidal cells at the equator undergo meiotic division, elongate and lose their nuclei and form the secondary lens fibers. The basal ends of the fibers remain attached to the basal lamina, while their apical ends added concentrically around the primary fibers, leading to the laminated appearance on the cut section. The increase in the equatorial diameter and enlargement of the lens with age is because of the continued production and persistence of new secondary lens fibers throughout life. The increase in the density during development is because successive fibers become tightly opposed and the layers become interdigited. The lens capsule, not a true lens capsule. Non-cellular envelope that completely surrounds the lens. Formed from the thickened basal lamina developed from the lens epithelium. Tunica vasculosa lentis, also known as vascular lens capsule. Vascular mesenchymal layer investing the lens, supplied by hyaloid artery, responsible for rapid growth of the lens during embryonic life. The anterior portion of this capsule is called the pupillary membrane. The tunica vasculosa lentis and pupillary membrane disappear during late fetal life due to degeneration of the part of the hyaloid artery supplying them. If persist, then it is known as persistent pupillary membrane. Lens sutures. A rect anterior Y-shaped suture has apex-to-apex apposition of the corresponding lens fibers from the two opposite sides near the anterior pole. Inverted posterior Y-shaped suture have base-to-base apposition of the detached lens fibers near the posterior pole. Blood supply of the developing lens is by hyaloid artery. Thereafter it depends on diffusion from aqueous humor and vitreous humor. The suspensory ligament of the lens. The mesenchyme, situated at the edge of the optic cup, giving rise to lens zonule. Congenital anomalies includes congenital cataract. Congenital aphakia, which is due to failure of development of lens placode during the 4th month caused by failure of its induction by optic vesicle and ectopia lentis.